Okay, happy children. This video is intended to be watched on Tuesday, December 14th, although I'm sure I will um, make some use in the future of recycling this, perhaps. Anyway, and it contains all the slides and in the guided notes that I was planning to do for Tuesday, but I'm going to be absent. Now, the nice thing about a YouTube video like this is if you are a slow note taker, say like Sydney, you can always pause the video whenever you want to take all the notes you need before I go on to the next slide. Whereas if you're a super fast note taker, like Lachlan maybe, you can just keep the video running. So anyway, let's get into the slides. Okay, so if you're watching this video, you presumably have a copy of the guided notes in front of you as well. And the guided notes will look very much like these slides, only that they have blanks in them. And again, you are welcome to pause and restart this video anytime you like to be able to take notes on what I'm saying. And so we're going to talk about cells, and so most of this video is going to be concerned with sort of the history of the microscope and the folks who devised what we call modern cell theory, and then a quick description of the difference between prokaryotic and eukaryotic cells, and then I'll cut it off there. Okay, so cells and cell organelles doing life's work. And so here are just some pictures of various cells. I'm thinking that these pictures on the left and right were taken using an electron microscope. This looks like a red blood cell that is part of a coagulation event, so perhaps part of a blood clot. This is a diagram of an animal cell with all of the organelles, and we will talk about that not in this video. So let's talk about who discovered the cells and who discovered cells. And to talk about who discovered cells, we have to sort of go back a little bit into the history of the microscope, because it was impossible to know that cells even existed before the invention of microscopes and the ability to see things that were very small. And so Robert Hooke was one of the first microscopists, and he made a compound microscope that had about 20 times magnification. So that's even less magnification than the lowest magnification on our modern compound microscopes. And he was looking at a piece of cork, and he saw what he called cells, and he called them cells. Now, I've heard a couple different explanations of this, because he saw little boxes in the cork and called them cells because they look like the rooms, that is, the cells at a monastery. There are also, I had read that he thought it looked like the cells in a honeycomb. And so the little six-sided things inside a honeycomb are also known as cells. Okay, here's a picture of Hooke, and this is his microscope. And and here is a picture of some pieces of cork. I think this is probably from his book, in which he was diagramming the cork cells. There's a better looking picture of his microscope on the next slide. And it's kind of an interesting device, because it's not exactly the same. It doesn't look exactly the same as our modern compound microscope, although it is a compound microscope. There is a lens in the objective, just like in our microscope. There was a lens in the eyepiece, just like in our microscope. And he had a screw here that would allow him to move the lenses closer and further from each other so that he could focus on the specimen. Over here, you have a kind of funky contraption, which is also shown over here in this photograph. And this was for producing light. So, of course, it was impossible to, I don't know if it was impossible, but they didn't have the kind of microscopes we have now that have light coming in from underneath through a slide. You just put the specimen in this little clamp, and you had to shine some pretty intense light on it. And so this is oil for an oil lamp. The actual flame, if you'll notice, is over here. What's this giant spherical flask full of water for? That is actually to take the light from this flame and focus it into this lens, and then from that lens was focused on the specimen. So this whole device was essentially a device for generating a very narrow and intense beam of light so that you could actually see what you were looking at. So that's the Hooke microscope. And so Hooke was the man. So his significance, what besides producing a book in which he illustrated hundreds and hundreds and hundreds of different cells that he was looking at, was also to coin the term cell. The first person to observe living cells, that would have been bacteria, sperm, blood, things like this, was Anton Van Leeuwenhoek. That's how I'm pronouncing his name. And he called them animalcules. Cule simply means diminutive, a small version of. So he thought they looked like small animals. And he was able to, rather than and Hooke, who had a microscope that was about 20-fold magnification, so there was a limit to what he could see. Van Leeuwenhoek came up with a microscope that actually had magnification greater than 200 times, and so that really allowed him to see a lot of small cells that were not possible to see before. And so this is a sort of a mock-up of his microscope. I frankly could not describe to you how this works because I don't get it by looking at it. And I saw a photograph of it online, and they really didn't describe how it worked either. But the big advance that he made was that he made a microscope that had much greater magnification potential than the one that Hooke was using, and therefore he was able to see actual living single cell organisms. Okay, so what's next? Well, we're going to talk about cell theory. So, having now observed cells and named cells and understood that single cells could be alive, then what happened next was the development of what we call cell theory, and this was between various scientists who were working with microscopes and observing cells and trying to make some decisions about what it all meant. And so, first of all, in the 19th century, there were a couple guys. Schwann was a zoologist, that is, a person who looks at animals. And so he used a microscope to look at animals, and in looking at over and over and over at numerous animals, came to the conclusion that all animals were made up of cells. Then you have Matthias Schleiden, who was a botanist, that is, he worked with plants. He looked at a lot of different plants, and he came to the conclusion that all plants were made up of cells. So you had Schwann reaching the conclusion that all animals, regardless of what the animal is, are ultimately composed of cells, and Schleiden reaching the conclusion that all plants, regardless of what plant it was, are ultimately composed of cells. And then it was Virchow, probably pronounced Virchow, but anyway, he also reported that every living thing is made up of these vital units, which we now know as cells. He was the first person to propose, well, maybe not the first person to propose, because I'd heard that Van Leeuwenhoek had also done this, that cells only come from other cells. In other words, cells cannot like spontaneously generate on their own. Every cell arises from the division of an existing cell. Okay, so let's talk about what cell theory tells us. There are three parts to cell theory, which, by the way, is why there's a little triangle here. The triangle of cell theory. There are three parts to cell theory. Part one, every living organism is made up of cells. This actually is a stain of some onion cells. So if you peel off a single layer of an onion, which is very, very thin, and look at it under a microscope, and these have actually been stained with a particular stain that stains that shows up things that are like proteins and nucleic acids. So that's why you're seeing the cell nucleus here. um, every organism is made of cells, and this example of an onion being a plant is made up of cells. So that's number one. Number two, the cell is the basic unit of structure and function in living organisms. And so you cannot, anything smaller than a cell is not alive on its own. So we have single-celled organisms like yeast, like amoebas that you've seen under the microscope, like bacteria. The cell is the smallest unit that can carry on life. So anything smaller than a cell, we wouldn't say that it is alive on its own. Okay, so the cell is the basic unit of structure. And then finally, a really key insight, as I mentioned, that was had in the 19th century and before, is that all cells come from pre-existing cell. And so the only way that a cell can arise is from division of an existing cell. There's no such thing as was once referred to as spontaneous generation. That is, that a cell could simply appear out of nowhere, or that just a murky set of chemicals would assemble itself into a cell. All cells come from division of existing cells. Okay, well, that is some history of microscopy, some history of the first observation of cells, the naming of cells, the first observation of living cells, and then the three parts of cell theory, which is that all animals and plants are made up of cells. The cell is the smallest unit of life, and all cells come from previously existing cells. Let me just quickly talk to you about the two types of cells. Well, here's a recap of cell theory. Let me talk to you briefly about the two types of cells. There are what are called prokaryotic cells, and a prokaryotic cell has no organelles. Now, we're going to talk not much in this video, but subsequently in talking about the guided notes, what we mean when we say organelles, but things that are inside the cell, like the nucleus, like the ribosomes, like the mitochondria, like other things like that, the Golgi apparatus. These are all what are called organelles. They are structures inside the cell that allow the cell to carry on its business. Prokaryotic cells do not have organelles. They're simply, all you can see here in this diagram of a bacteria is just a bunch of DNA floating around encapsulated inside a cell. And this window that you can see in the cell doesn't actually exist in any particular cell. The artist has drawn a little window in it, just like the artist has kind of cut away the cell here and cut away the cell here to allow you to see what's inside. So inside a prokaryotic cell is just some DNA floating around encapsulated in a cell membrane. Eukaryotic cells, and bacteria cells are examples of prokaryotic cells. The other type of cell is called a eukaryotic cell, and a eukaryotic cell contains organelles. So, for example, in this animal cell, here's the nucleus. In this plant cell, here's the nucleus. In this animal cell, you can see what's called the endoplasmic reticulum here. You can also see it in the plant cell. In the animal cell, you can see some mitochondria here. You can also see it in the plant cell. Okay. All animal cells, and all plant cells are eukaryotic cells. In fact, any multicellular organism is made up exclusively of eukaryotic cells. So humans, dogs, cats, rats, mice, even down to some single cell things like yeast and paramecia are all made up of eukaryotic cells, and all plants are made up of eukaryotic cells. Only bacteria are prokaryotic cells, and another subdivision of cells known as archaea. But archaea and bacteria are very closely related to each other. We will mostly focus our discussion of cells in this unit on eukaryotic cells because they contain organelles, and those are the things we're going to be learning. There's a distinct difference in the size of prokaryotic cells and eukaryotic cells. Prokaryotic cells are much, much smaller than eukaryotic cells, and that makes a lot of sense because all they have to contain really is this string of DNA and just a few other things floating around inside what's called the cytoplasm. Whereas a eukaryotic cell has to contain a nucleus and various other organelles. And so typically a eukaryotic cell is 10 to 100 times larger than a prokaryotic cell. And so they're kind of trying to show this to you here, that this bacterial cell, by comparison to a eukaryotic cell, it looks a little bit like a spaceship orbiting around the Earth. And so most bacteria are in the range of 1 to 10 microns in diameter, whereas most eukaryotic cells are 10 to 100 microns in diameter. And a micron is 1/1,000,000th of a meter, or 1/1000th of a millimeter. Okay, that's as much as I am going to include in this video. So thank you very much for watching.